Yes, it's titled Biological Classification, the Five Kingdom Master Guide.
Yeah.
Okay, let's unpack this, because looking at this progression, it isn't just a list of scientific names. It is essentially a historical blueprint of human beings wrestling with the sheer scale of nature.
Trying to put everything from, microscopic bacteria to the massive blue whale into boxes that actually make sense.
Exactly.
It really is a story of human perception evolving. And if we connect this to the bigger picture, you have to realize why classification or taxonomy matters so much to begin with.
Oh, absolutely.
Imagine walking into a library where millions of books spanning thousands of genres have just been dumped in a massive pile in the center of the room.
You wouldn't be able to find a single thing.
Not a chance. The diversity of life on Earth is exactly like that pile. Without a rigorous, standardized system, studying life is just overwhelming chaos. Classification is our ultimate quest to find patterns, to track origins, and to understand the deep evolutionary story that connects us all.
And that quest starts on page 2 of our guide with the grandfather of observation, Aristotle. His ancient system was the first real attempt to build some shelves in that chaotic library.
Build some shelves, yeah, that's a good way to put it.
But his method was incredibly simple, almost comical to us now. For plants, he just divided them by size, trees, shrubs, and herbs.
Very scientific.
Right. And for animals, he looked at one single metric: animals with red blood and animals with no red blood. I mean, you can picture him just squishing a beetle, seeing clear fluid instead of red blood, and deciding, well, this goes in a completely different category from a dog.
Which, to be fair, is entirely relatable from a purely observational standpoint.
Sure.
He was working exclusively with the naked eye and immediate physical traits. But of course, as our understanding grew, that system fell apart completely. You can't categorize the world just by what bleeds red.
No, you definitely can't.
That realization eventually brings us to Carolus Linnaeus and his two-kingdom system. He gave us Kingdom Plantae and Kingdom Animalia. It was a step forward, sure, but it had massive glaring drawbacks that honestly held science back for a long time.
Right, because Linnaeus basically said, if it moves and eats, it's an animal. If it just sits there and grows, it's a plant.
Exactly.
Which led to some absurd groupings. He lumped prokaryotes, like bacteria which don't even have a true nucleus, into the same overarching categories as highly complex multicellular eukaryotes.
It was a mess, and the most egregious error was lumping fungi together with green plants. Think about the fundamental biological difference there.
It's huge.
You have a mushroom, which is a heterotroph that has to actively consume and break down decaying organic matter for fuel, and you're putting it in the exact same box as a fern.
A fern, which is an autotroph that literally builds its own food out of sunlight and carbon dioxide.
Exactly. Morphologically, they both grow out of the ground, but metabolically, putting them in the same kingdom is like saying a solar panel and a gas-powered generator are the same machine just because they both sit in your backyard.
That is a perfect way to put it. The categories were just too broad to be biologically meaningful, which sets the stage beautifully for page 3, the modern solution.
The big shift.
In 1969, a scientist named R.H. Whittaker proposed a massive breakthrough, the five-kingdom system. He finally broke life down into Monera, Protista, Fungi, Plantae, and Animalia.
Whittaker didn't just guess at these divisions either. He built this system on 5 crucial, incredibly rigorous criteria.
Let's hear them.
First, cell structure. Are they prokaryotic or eukaryotic? Second, body organization or the phallus. Are they single-celled or do they form specialized tissues and organs?
Yeah.
Third, mode of nutrition. Fourth, reproduction. And fifth, crucially, phylogenetic relationships.
Wait, let's pause on that last one. Phylogeny basically means evolutionary history, right?
That's right.
How was he able to confidently map out evolutionary relationships in 1969 compared to someone like Linnaeus centuries earlier?
That is the key difference. By the mid-20th century, we had electron microscopes, a much deeper understanding of genetics, and better fossil records.
Oh, of course. The technology caught up.
Exactly. Whittaker wasn't just classifying organisms by how they look today. He was classifying them by who their ancient ancestors were. He was building a family tree, not just sorting a pile of rocks.
And here's where it gets really interesting on page 4. If you map all 5 of Whittaker's kingdoms out side by side in this master blueprint, a really strange, elegant evolutionary story appears, especially when you look at the architecture of the cells themselves.
It's brilliant.
Kingdom Monera stands completely alone across the entire board. It is the only kingdom made up entirely of prokaryotic cells. Because of that, it is the only kingdom with absolutely no nuclear membrane.
It is a profound isolation. In Monera, the genetic material isn't safely locked away in a central vault like it is in our cells.
Exactly. So does their DNA just float around freely like loose spaghetti? How does a cell even function like that without it all unraveling?
It does float freely, but it's organized into a dense central region called a nucleoid.
Nucleoids.
Yeah, it's essentially a coiled-up clump of DNA. It's highly efficient for rapid replication, which is why bacteria can multiply so incredibly fast. They don't have to break down a nuclear envelope every time they divide.
That makes a lot of sense.
And if you track the evolution of the protective cell wall across Whittaker's kingdoms, you see another brilliant pattern in this chart.
Yes. In Monera, the cell wall is non-cellulosic. It's built from amino acids and sugars. Then you step up to Kingdom Protista, where a cell wall is only present in some organisms.
Yeah.
Step sideways to fungi, and suddenly the cell wall is made of a tough, leathery substance called chitin, the exact same stuff you find in crab shells.
Which is wise.
Move over to Kingdom Plantae, and it's made of cellulose, allowing trees to grow hundreds of feet tall. And finally, when you arrive at Animalia, the cell wall completely vanishes.
And that disappearance is the very reason you and I can move, dance, and breathe. If our cells had rigid walls like a plant, we would be entirely immobile.
We'd just be stuck there.
We traded the structural armor of a cell wall for the flexibility of a simple cell membrane, enabling locomotion and complex nervous systems.
I love how one microscopic structural change completely dictates how an organism experiences the world. So, having established this master blueprint, let's scale all the way down to page 5 and enter the microscopic realms. Starting with Kingdom Monera, the bacterial world.
The survivors.
Truly, these are the most abundant microorganisms on Earth. They're everywhere. You find them in boiling hot springs, at the bottom of crushing deep ocean trenches, and buried in polar snow.
Their adaptability is unmatched in the history of the planet. And while they might just look like specks under a basic microscope, structurally they fall into 4 distinct, highly functional shapes.
Right. You have coccus, which are spherical, like tiny berries. You have bacillus, which are rod-shaped, often carrying resilient little spores inside them.
Tough ones.
Then there's vibrio, which look exactly like commas. And finally, scirillum, which are spiral-shaped and usually have these long, whip-like tails called flagella, acting like upward motors.
But within this kingdom, there is a massive evolutionary divide on page 6. We have to talk about the extremists, the archaebacteria.
Oh, the rule breakers.
The archaebacteria are ancient. They possess a fundamentally different cell wall structure from regular bacteria, which acts almost like a microscopic hazmat suit.
A hazmat suit, that's a great image.
It allows them to thrive where literally any other form of life would instantly dissolve or fry. You have halophiles thriving in insanely salty environments like the Dead Sea. You have thermoacidophiles, which actively love boiling acidic hot springs.
And then you have the methanogens, which prefer marshy anaerobic environments. And here is a wild, real-world connection. Methanogens actually live in the guts of ruminant animals like cows.
Yes, they do.
They help break down the tough cellulose the cows eat, and in the process, they produce methane gas. So when you read articles about cattle farming and cow burps driving global warming through greenhouse gases, you are literally reading about the metabolic byproduct of archaebacteria.
It's the perfect illustration of how microscopic life directly shapes massive macroscopic global systems. The atmosphere of our planet is largely dictated by organisms we can't even see.
It's wild.
Now, the other side of Monera is the eubacteria, the true bacteria. These have rigid cell walls and include things like cyanobacteria, often called blue-green algae.
Right.
Cyanobacteria are autotrophic. They have chlorophyll A, meaning they photosynthesize. Billions of years ago, they were the ones responsible for pumping the first major waves of oxygen into Earth's toxic atmosphere.
But eubacteria also include the vast army of heterotrophic bacteria. These are the decomposers. And honestly, they are arguably the most ecologically critical organisms alive.
Yeah, absolutely.
Without them breaking down dead matter and recycling carbon and nitrogen back into the soil, the entire global food web would collapse in a matter of weeks. The world would just pile up with dead organic material.
We owe our entire existence to their janitorial work.
Which is a humbling thought. Now, stepping up the evolutionary ladder to page 7, we leave the prokaryotes behind and enter Kingdom Protista. This is the great aquatic link.
The bridge.
Protista is fascinating because these organisms are single-celled, just like bacteria, but they are eukaryotes. They finally have a well-defined nucleus. They form this crucial evolutionary bridge connecting the simple bacteria to the highly complex plants, animals, and fungi.
And what's fascinating here is the sheer dramatic contrast and variety housed within this single kingdom. They are the true crossroads of evolution.
You have groups like the slime molds, which act like slowly creeping, pulsating decomposers in wet forests.
But let's look at the oceans on page 8, specifically the chrysophytes, commonly known as diatoms.
Diatoms are incredible. They are microscopic plankton that act as the chief producers in the oceans, doing a massive amount of the Earth's photosynthesis. But structurally, they are stunning.
They really
They
are.
build incredibly unique siliceous cell walls that fit together exactly like a tiny, indestructible glass soapbox.
And because that glass-like silica is virtually indestructible, when diatoms die, their shells don't decay, they sink.
Right to the bottom.
Over millions of years, they accumulate on the ocean floor in layers hundreds of feet thick, creating something called diatomaceous earth. It's an incredibly gritty, porous, and durable substance.
Humans actually mine ancient dried-up ocean beds to harvest it. We use it for industrial filtration, soundproofing, and even as a fine polishing powder. It's wild to think that when you polish metal, you might be using the crushed glass houses of ancient microscopic ocean life.
That's the beauty of Protista, but they aren't all benign glass houses. On the other end of the spectrum, you have the dinoflagellates. These have two flagella, allowing them to spin as they move. Take a specific one called gonulax. Under certain nutrient-rich conditions, gonulax will multiply so rapidly that it triggers an algal bloom. The sheer concentration of their pigments literally turns the coastal seas blood red.
The infamous red tides. And this isn't just a pretty color change. If you've ever lived near a coast during a severe red tide, it is a visceral, awful experience.
Very toxic.
They release highly potent neurotoxins into the water. The beach becomes littered with thousands of dead fish, and the waves actually aerosolize the toxins. If you stand on the beach, it makes you cough and burns your throat. It's a striking reminder of the devastating power these microscopic, single-celled organisms wield.
Absolutely. And that devastating power extends beyond the oceans and right into our own bloodstream. Moving to page 9, within Protista, there is a group called the protozoans.
The predators.
These are heterotrophic predators or parasites, and evolutionary biologists believe they're the primitive single-celled relatives of animals.
One group of protozoans, the sporozoans, really demands our attention here. These organisms have a highly infectious, spore-like stage in their life cycle. The prime example is Plasmodium.
The parasite responsible for malaria.
Exactly.
It is practically impossible to overstate the historical weight of Plasmodium. This isn't just a biological curiosity, it has had a staggering, tragic effect on human history. It has toppled empires, halted armies, and fundamentally altered human genetics in certain regions.
Millions upon millions of lives have been lost because this primitive relative of animals found a way to use a mosquito to hijack the human bloodstream.
Understanding this classification system isn't just academic, it is the foundation of global public health.
It really puts the stakes into perspective. Okay, moving forward to page 10, we finally leave the exclusively single-celled microscopic world behind and look at the macro world. Let's talk about kingdom fungi, the great recyclers of the earth.
The decomposer.
You mentioned earlier that fungi are heterotrophic. Specifically, they act as saprophytes, meaning they secrete enzymes to dissolve organic matter from dead substrates outside their bodies and then absorb the nutrient soup. Or they act as parasites.
Right.
We know their cell walls are made of chitin. But there is a glaring exception to the multicellular rule here, yeast.
Yeast is always the outlier. It's a completely unicellular fungus, which makes it incredibly useful for baking and brewing, but it often confuses people who think all fungi are just mushrooms.
Right. Now, for the multicellular fungi, their physical architecture is so alien compared to ours. They don't have tissues or organs.
No.
They're built from long, slender, thread-like structures called hyphae. When thousands of these hyphae weave and network together, they form a massive web-like structure called a mycelium. Some of these underground mycelium networks are miles across, making them the largest living organisms on Earth.
And if you look closely at those individual hyphae threads, you see two distinct structural strategies. You have coenocytic hyphae and septate hyphae.
Let's use an analogy here. Think of coenocytic hyphae as an open concept house.
Yeah.
It's one long, continuous hollow tube filled with cytoplasm, and all the nuclei just share that massive open space. There are no walls. Septate hyphae, on the other hand, are like a house with a bunch of individual rooms. They have cross walls or septate dividing that long tube into distinct cellular compartments.
That's a great way to visualize it. And that unique architecture plays a huge role in how they reproduce on page 11. Fungi can reproduce vegetatively by simply breaking off a piece of hyphae in fragmentation or through asexual spores. But their sexual reproduction cycle is a truly bizarre three-step evolutionary dance.
Okay, walk us through this dance, because it is very different from animal reproduction.
It is. Step 1 is called plasmogamy. Imagine two different fungal hyphae meet underground. During plasmogamy, the actual cellular contents, the protoplasms, fuse together.
Like the house is merging.
Going back to your house analogy, exactly. It's like two neighboring houses knocking down their shared walls so the living spaces merge. But here's the catch. The nuclei from the two parents don't fuse yet. They just share the same room.
So the families have moved in together, but they are still keeping completely to themselves.
Exactly. Sometimes they stay in this weird intermediate phase for a long time, but eventually, step 2 triggers karyogamy.
Karyogamy.
This is the actual fusion of those two parent nuclei, finally intermarrying and creating a diploid zygote containing a full set of paired chromosomes, and then immediately, step 3 occurs, meiosis.
Split.
The zygote undergoes division to cut that genetic material in half, resulting in haploid spores. These spores are eventually pushed up and released by specialized structures we call fruiting bodies, which are the actual mushroom caps you see popping up out of the dirt after a rainstorm.
The mushroom is just the delivery mechanism for the spores. It's brilliant. From the decomposers, we make a massive shift over to page 12. The producers. Kingdom Plantae.
The green world.
Biologically speaking, they are eukaryotic, multicellular, and contain chlorophyll, allowing them to harness solar energy. They have those rigid cellulosic cell walls we talked about. But nature hates a strict rule, so we have to highlight the rebels. Not all plants are purely autotrophic.
No, evolution always finds a loophole. You have insectivorous plants like the Venus flytrap or the pitcher plant. They live in soil that is drastically poor in nitrogen, so they evolve to catch, kill, and digest insects to supplement their diet.
Carnivorous plants.
And then you have the true parasitic plants like Cuscuta or Dodder. It looks like a tangled mess of yellow spaghetti. It has no chlorophyll of its own, so it literally wraps around a host plant, pierces its stem, and vampirically drains its nutrients.
Vampire plants.
Mm-hmm.
But perhaps the most conceptually mind-bending part of Kingdom Plantae is their life cycle, known as the alternation of generation. Plant life cycles are not a straight line from parent to child like ours. They swing like a pendulum between two totally distinct, alternating biological phases.
Right. You have the diploid sporophyte phase. This phase has two sets of chromosomes, and its entire job is to produce spores. Those spores don't grow into another sporophyte.
This is the weird part.
Instead, they land and grow into an entirely different organism called the haploid gamophyte phase, which only has one set of chromosomes. The gamophyte's job is to produce gametes, sperm, and eggs. When those gametes fuse, they grow back into a sporophyte. It's a continuous generational relay race.
To put that into perspective, imagine if humans reproduce this way. It would be like you giving birth to a completely different intermediate species. Let's say a small, green, goblin-like creature.
A goblin.
And that creature lives its own life and eventually gives birth to a normal human again. That is what plants are doing every single generation. It's wild.
It really is.
Now, let's bring it all home to page 13, our own territory. Kingdom Animalia, the consumers. We are eukaryotic, multicellular, and we have absolutely no cell walls. Because we completely lack chlorophyll, we're relying entirely on plants for our energy, either directly by eating them or indirectly by eating something that ate them.
And what defines us goes beyond just what we eat. It's how we eat. Animals utilize holozoic nutrition. This is a major evolutionary leap.
How so?
Instead of secreting enzymes outside our bodies to dissolve food in the dirt like fungi, we actively ingest our food whole, internalize it, and digest it safely inside a specialized internal cavity. We then store our excess energy reserves highly efficiently as glycogen or fat.
We also have a very different growth strategy. A tree will continue to grow from its branch tips indefinitely as long as it lives, but animals have a definite growth pattern. We grow into a highly specific, genetically predetermined shape and size, and then we stop.
And because we aren't rooted to the ground, we face entirely different survival challenges. To navigate a three-dimensional environment, hunt for food, and escape predators, higher forms of animals had to evolve immensely complex sensory and neuromotor mechanisms.
The brain.
The leap to active locomotion required a brain that could process visual, auditory, and olfactory data in milliseconds. It completely changed the trajectory of biological complexity, leading eventually to conscious thought.
It is a phenomenal system. So Whittaker gives us these 5 kingdoms. We have life perfectly sorted, classified, and boxed up.
Or so we thought.
But Biology is never that clean. On page 14, just when you think the rules are set, you encounter the ultimate plot twist. The outliers, the entities that completely defy Whittaker's criteria. Entities that, according to traditional scientific definitions, aren't even truly living. We are talking about viruses, spheroids, and prions.
This is where biological classification genuinely breaks down. Let's start with viruses. They aren't made of cells, they have no metabolism, they don't consume energy, and they cannot reproduce on their own.
So what are they?
They are just incredibly minimal nucleoproteins, a protective geometric protein coat called a capsid, wrapping around a tiny strip of genetic material, either RNA or DNA. They're basically rogue genetic code in a tough envelope.
A tough envelope.
They just float around until they bump into a living cell, inject their code, and completely hijack the host cell's machinery to print thousands of copies of themselves, usually destroying the cell in the process.
They are the ultimate biological hackers. But if you thought viruses were minimal, wait until you hear about viroids. Discovered in 1971 by T.O. Diener, these things are just strands of free, naked RNA.
Completely naked?
They completely lack the protein coat. They are significantly smaller than viruses, yet this naked floating piece of code is famous for causing potato spindle tuber disease, utterly devastating agricultural crops. Just a tiny snippet of RNA causing all that systemic damage.
It is deeply unsettling how something so simplistic, something lacking every hallmark of life, can be so destructive. And that theme gets even darker on page 15 with prions. If a virus is genetic material in a coat, and a viroid is naked genetic material.
A prion has absolutely no genetic material at all. Zero DNA. Zero RNA.
Right.
How does it even exist then?
It is simply a protein that has folded into an abnormal pathological shape. They are similar in size to viruses. But here is the terrifying part. When a prion bumps into a perfectly normal, healthy protein in a host's brain, its bizarre shape somehow induces that normal protein to spontaneously refold and become abnormal, too.
It's a chain reaction. It's literally like a zombie protein turning healthy proteins into zombies just by touching them.
Exactly. And because they are just misfolded proteins, the host's immune system doesn't recognize them as an invader. This slow, unstoppable chain reaction causes severe, incurable neurological diseases.
Like mad cow disease.
Bovine spongiform encephalopathy, yes. If you remember the absolute panic in the agricultural world in the 1990s, it was because prions break all the rules of infection. You can't kill them with standard sterilization or cooking because they aren't alive to begin with. They are just a shape.
That is legitimately terrifying. To recover from zombie proteins, let's end on a much more harmonious outlier. Lichens. If you've ever seen crusty, colorful patches growing on a boulder or tree trunk, that's a lichen. But a lichen isn't a single organism. It is the ultimate symbiotic relationship. It is two completely different organisms living seamlessly as one.
It is a beautiful example of mutualism. You have an algae component called the phycobiont. The algae contains chlorophyll, so it photosynthesizes and provides the food.
And wrapping around that algae is a fungi component.
The mycobiont. The fungi provides a tough structural shelter, prevents the algae from drying out, and absorbs water and minerals from the rock. Neither could survive on that bare rock alone, but together they conquer it.
And they serve a vital ecological function for us too. Lichens are incredibly sensitive to air quality, specifically sulfur dioxide. They are excellent biological pollution indicators because they absolutely will not grow in heavily polluted industrialized areas.
They need clean air.
So if you are hiking and you see a forest beautifully draped in thick, healthy lichens, you can take a deep breath, knowing you are standing in genuinely clean air. So what does this all mean? We just traveled sequentially through the entire blueprint of biological classification.
From page 1 to 15.
We watch humanity evolve from Aristotle sorting bugs by blood color, to Linnaeus fumbling with plant-like animals, all the way to Whittaker finally mapping the deep evolutionary family tree. We've marveled at perfectly folded prions causing neurological chaos, microscopic diatoms leaving behind vast glass oceans, and the bizarre pendulum swing of plant reproduction. It's a profound testament to the bewildering complexity of nature and our stubborn, beautiful human desire to make sense of it all.
It certainly is. But looking at where our journey ended, this raises an important question for you to ponder.
Okay, let's hear it.
We spent all this time outlining the rigorous, complex criteria required to be classified as living in the five kingdoms. Yet, we ended with the absolute outliers, viruses, viroids, and prions. These entities completely lack the basic biological criteria to be classified as alive, yet they possess the extraordinary power to hijack, fundamentally alter, or completely destroy the highly complex organisms that are in those kingdoms. If non-living things can command and dismantle the living so effortlessly, does humanity's very definition of living need an evolutionary upgrade?
Now that is something that will definitely keep you thinking long after this is over. Thank you so much for joining us on this deep dive into the blueprint of life. Keep exploring the world around you, question the categories we put things in, and most importantly, stay curious.